to India and then joined, higher education for a few months, and became the director there, Department of Chemistry, and of course he taught physics also. Then again come back to IIT Delhi. Then he goes to Canada as postdoc. Then he goes to Cambridge and for his PhD work. And then he joins Cambridge, in a managerial post. So I found very interesting that a scientist is not picking up accountancy, but he has done also. So he's a man of all seasons. And then, Mississippi State University, University of Medical Center in the Netherlands, then in Hanover, Germany, and finally he is in Virginia Tech. See, he's going to talk about multi-omics. Omics and omics, as you all know, has been a subject of a lot of interest, because everybody knows proteome. See. But when you come to proteomics, then what is proteomics? So naturally we all know that proteome is a protein that is produced by the cell or a tissue or organism, the total. And proteomics would be when you are hitting at a particular protein, trying to identify it, locate it, and see how it really works in the system. So similarly, we have transcriptomics, and I was looking at the details. There are 40 such omics and omics. So he will be talking about this multi-omics driven life sciences. So without taking any further time, I'm great honor and pleasure becoming Dr. Abhishek Singh, Abhishek Narain Singh. See. So it is all yours now. Thank you.
Thank you, Professor Bhargav, for a very humble introduction. Thank you, everyone, eminent faculty and staff, for taking out your precious time for attending this brief presentation. As already spoken by Professor Bhargav, I'll be talking about multi-omics, which is a new buzzword in the scientific analytics domain, where an integrated approach is needed for solving grand challenges of scientific problems, of solving terabytes, petabytes of data. The age that we are moving in, we require a multidisciplinary combination of computer science, other sciences, and biotechnology and life sciences definitely as a combination to solve the problems and come up with something interesting. So with this I start off with my talk, multi-omics driven life sciences discoveries and inventions. So I'll be picking up the metabolomics first and cell signaling. So as we know, I don't know if the pointer reaches there. Okay, it does reach. So as we know that if you know CEMS, capillary electrophoresis mass spectrometry, or multi-MS technology, we have a long peptide that is being cleaved at certain location by means of targeted enzyme, and that smaller peptide now that is generated, that is then charged, and the charge to mass ratio or mass to charge value, whatever the case be, is then recorded. And this is done repeatedly for different dilutions of the solvent, and what we obtain is some kind of a random plot. So typically, for example, if someone is suffering from disease which is at that stage generates a protein which can be a signature for the disease, then what we can do, a bodily fluid such as from the urine or maybe even the blood sample, that can be extracted, and then we can filter out and carry out this mechanism and look for the signatures of the peptides that are already known in the literature, scientific experimental literature, to have had record of a known disease, or either directly or in some way having an effect. So the challenge as an informatics person would be that we see that any of these plots, they are not so regular. None of the plots, all of them show a downward slope. None of them, all of them show an upward slope. There can be data points which reaches a near zero. There can be data points which is a near zero in the beginning, such as the blue line over here. So the algorithm would be pretty simple, but I didn't know why did anyone think about it. This was the work that I did as a Marie Curie experienced researcher while I was in Germany. So it was called the Protoclin project. And what I simply propose is that what we do is simply get rid of the noises that show a deviation from what should be there, either before or after, and consider each of the curve on a case-by-case basis. So what a MATLAB version code would be, if this is one of the curve, the blue line, then a multi-regression line would be this red line, and after I reduce the noises, such as the ones in the end and the ones in the front, until we get a traditional slope or a traditional pattern, then we get this green line as the modified curve. So you see such a great difference in the slope of S, and it can make a big difference in the predictions that are made eventually. We can also go for a non-linear regression if needed. So eventually I made, well, MATLAB is a paid software, so we want to go for an open technology such as Java. So I made a Java standalone application for that. So this was the metabolomics part. We want to know how this can be affecting the biochemical pathways, such as in developmental biology. How does a cell, starting from a single cell, takes up a shape of its parent? For example, a single cell of a lizard eventually takes up the shape of a lizard. A single cell of a human being eventually takes up a shape of a human being. And if this whole developmental pathway goes wrong, then what we can end up is some kind of a cancer happening. So this is the kind of metabolomics study that we want to know, that we know that there are interconnected bioreactions happening with their own mathematical models. I think all of us are educated, we know of it. Kinetic forward and kinetic backward reactions, which are all connected, and we can put in some kind of a signal that is coming out of the cell. So it's essentially what happens is this whole unit is a virtual cell which can be controlled by us, whether in a bioreactor, or if it is successful, we can even transform the reaction later on to living human beings. So the key concern is, how does this signal makes this whole reaction trigger on eventually. It's not the amount of the external signal, but it's actually the delta value, the change in the amount of signal that influences the onset of the whole reaction. So modeling it in a chemical kinetic pathway, so this is what the initial stage is, where the initial signal is 0 with time. This is how the components vary. If I change the signal from 0 to 1, representing a significant change in the external condition of the compound which is triggered the reaction, we can essentially depict how each of these components, coming back to the reactions again, like beta-catenin, which is important, changes with time, and that can give us answers. How do different organisms develop in different ways? How do certain onset of cancer take place? And if once we have a control of this cellular environment in a mathematical way, this was confirmed by, this work was done in Los Alamos labs, an immunological group, and we had collaborations from Harvard that eventually confirmed that this model that we made mathematically for the Venn signaling pathway was predicting to be absolutely correct with marginal error of 0.01%. So that's what it is. I mean, you have mathematics, you have computer science, you have knowledge of biological sciences, and you have to apply software to integrate these things together. So talking about metabolomics pathway once again, such as the flux change, we know if there's a reaction A plus B giving a compound C, which reacts again with D, and so on and so forth till Z. And if these downstream compound can bind to the protein P or enzyme EP at a certain pocket known as the allosteric domain pocket, which affects the catalytic active site pocket, either feed forward activating it, feed forward deactivating it, increasing it tenfold or decreasing it tenfold. All kinds of possibilities are there. These compounds can also be, before these reactions. So we, in a way, the evolution has happened in order to have a controlled circuit. And for example, if Z is a precious compound, such as vitamin B5, or say G is vitamin B5 and Z is an even more precious compound downstream of vitamin B5 coenzyme A pathway, such as the biofuel lipid pathway. So what we want is that we want to cut short all these circuits, and we have more of these Z produced or B produced as the case, G produced as the case be. So one of the hypotheses could be that you've built in some kind of a membrane here, so that which is selectively permeable. C can go out, but none of these components can get in. So in that way, none of the downstream product or maybe even the upstream product can interact with P, and we only have thus the active sites participating in the reaction and not the allosteric domain. However, this logic can kind of fail in a biological cellular environment, because a cell will divide and daughter cells will be produced, and then those will divide and another daughter cells will be produced, and if we have control that this membrane will also divide in the same proportion and get carried on, inherited to the daughter cells, then this logic works. But if it doesn't, then this reaction can work only for traditional chemical reactions, not for biochemical reactions. So what can be proposed is that we keep this membrane for 1 million years, or maybe 500,000 years as the case be, and then we can depend on evolution. We can say that since the D downstream product are no more interacting with the allosteric domain, since the functionality of the allosteric domain is no more needed, all the mutations that happen in the enzyme pocket of allosteric domain, that will die off, that will kill off with time. And so after 1 million years, if I remove this membrane, then we will have an uncontrolled, or rather in our ways, controlled reaction. So this was the kind of a patent that was filed. This kind of research was done when I was in Cambridge University, when I was working on vitamin B5 pathway regulation, or rather deregulation. But this concept can also be applied for cancer, tumor cure pathway, when we know that this is overproduced and the reaction is going out of our control, unwanted proteins are being synthesized, we can adopt a similar concept over here. So as an example, if I go to vitamin B5 pathway, I'm pretty sure many of you guys must be aware of this. So we have this reaction happening, and these are the intermediate metabolite, and these can bind to this pocket over here, pantothenate, which is vitamin B, pantothenate, L-pantosate, and this can all bind to certain pockets over here and seize the onset, the carry on of the reaction. So the concept would be just making a blocking over here, a pocket, and wait for 1 million years. Now the obvious question that must be coming to your mind is that why wait for 1 million years? So obviously that's not the solution. That's the theoretical part of what Darwin's theory of evolution is, by the survival of the fittest. But a more practical solution to this would be that we do a site-directed mutagenesis, SDM technology, and get things done faster. Or what can be done is that instead of going forward 1 million years, we can go phylogenetically 1 million years backward, looking for a similar pathway in other organisms where there are compartments present. So this way that this can be, technology can be adapted. So coming to genomics, this is the most interesting area which has caught attention of most of the scientists worldwide. So for example, if any two individuals, random individuals are selected, then we all are similar by more than 99%. And that's why if anyone is given a training to do something, he's able to do it, because any two person are randomly not more than 99% similar. However, there are minute differences which can be attributed to the differences in which how people react, how people behave, how people respond to drugs, what are the different traits, and those are the minute differences that are important to capture. So if I have two random individuals, say myself and any person X, and I compare the two genome is a set of all DNA, which is typically of the order of 3 gigabytes for human beings. So then the variations can be structural variation or single nucleotide polymorphism, and the structural variations can be insertions or deletions, inversions and copy number variations, which can be further subcategorized, whether they are intrachromosomal or interchromosomal and truly novel or really novel. Those kinds of classifications are being made. So this is the kind of plot that we are interested in, some kind of a database that is generated when a country of 6 billion or 7 billion population are there. We want to make a really personalized approach towards dealing with everyone. This is the kind of a chip that everyone needs to carry, which will have this variation information. So for example, if I'll make it really short, I wouldn't get into the details of what the algorithm is. The current sequencers are not capable of sequencing long sequences. Well, in other words, they are capable of sequencing long sequences, but the quality of the confidence that we have in every basis, that typically drops off after certain lengths. And so that's the reason sequences are limited to certain lengths where we have certain confidence in the number of, in the bases that are being generated. So, say for example, the current generations, you can sequence it to 200 bases, and if you go beyond, you might find a polygenetic degradation to a great extent. So if we have a front end, we have a back end, and we have a knowledge of rough insert size in between, and then we take the two pairs and map it to the genome, and then we see that the insert size that we estimated to be, say, 400 bases is not coming out to be what we thought. Then there can be an insertions or deletions over there. So for example, if it exactly matches what we expected, then there is no insertions deletions here. If it doesn't match, there can be insertions deletion here. Similarly, there can be duplications if we sequence a genome n number of times, and then once we map it back, the sequences to the genome, it can be maybe 2n number of times. So we can say that this certain region of a genome, which can be a gene regulation region, or maybe a gene dense region itself, or a cascade, then that can, by the read depth itself, we can confirm the copy number variations that can exist. And then similarly, if it doesn't matches, we have a deletions, and split read, as the figure says, the reads are split at certain region, and we have more such reads doing it. So essentially, I made software called MSECT, Passive String Exhaustive Comparison Tool, and Genome Break. MSECT is more computationally intensive than Genome Break, but it gives you more results. MSECT is capable of capturing copy number variations as well, that I mentioned to you on the previous slide. So what we have is a reference genome, and this is one of the person whom we are interested. This is 3 into 10 to the power 9 roughly number of bases. That's the order of data that we are typically dealing with. And when we make a plot of the comparison with different intensities, we see that levels of match, the percentage identity we get over here. So it's data intensive, it's compute intensive, and you have to do it in a very efficient, robust manner. And genome break, you see the diagonal line over here, that's what is generated by the genome break, and that is typically good in extracting insertions, deletions, and SNPs of our interest. That's where the research over the world is going forward at this stage. People are focusing mostly on insertions, deletions, and SNPs at the moment. So what are the case be? Once that is there, we can, each loci of our interest can then be explored to where the matches exist. And as I said earlier, that before the human genome draft was even prepared, we thought that the genomes are 99.9% similar. After the genome draft was made available, I was working in Toronto at that time, and our group published that the genomes are likely less than 99.5%, about 99.5% similar. So the number of variations that we expected out of 2 random individuals now increased by a significant amount. However, I later did a reanalysis, and I published that this amount can be even less than 99.5% similar. And this is the variation that contributes to one person getting ill and the other person not getting ill under the same exposure of environment. One person getting cured much quicker by a certain drug, the other person taking several months to get cured for the same disease by the same drug. And this is the reason why certain families are observed to have offspring or children which are taller, and other families are observed to have children which are fatter. So these are like the broad classification, but this classification can go much into deeper roots, such as we can go into diseases like autism. There has been genetic linkage, DNA linkage of why autism occurs and to what extent. ADHD disease or disorder, I would say. Major depression or so these are all mental sickness that I mentioned here, but it can also be like schizophrenia, brain tumor, so bipolar disorder. But they can also be extended to other diseases such as diabetes or any like sickle cell anemia. These are all already characterized for having their genetic roots. So essentially what we have is a medical informatics person, one of us, having several individuals coming to them with their genome chip and the medical informatics person using a supercomputer or maybe cloud computing, great computing these days, might be able to classify people based on the kind of disease or the kind of characteristics, kind of traits that we're looking for. So one of the things that I did when I was in Holland and Amsterdam was took an Amsterdam population of family of 4, which had the father, A105A, B is a mother, A105B is a mother, and the 2 daughters. And I could simply look at the